All right, so our next speaker is Teresa McGee from UNC Chapel Hill. She's a grad student in the Valdar lab and studying gene bioenvironment interactions, particularly with hepatic liver, functionality.
Just a quick mic check. Can you guys hear me pretty well? Okay, cool. So my talk is actually, I think, fairly fit for this conference, given Rob's introduction on Monday, where he emphasized the needs for diverse models in clinical testing and expanding out our rat and mouse resources. So I'm really excited to talk to you guys about this work that we've been working on for quite a good amount of time in the lab, where we are hoping to characterize the collaborative cross as a resource for pharmacogenetics. And my study in particular, what I'm focusing on today, is characterizing the drug metabolism, enzyme, and transporters in the liver of these collaborative cross mice. And from here on out, I'm not going to say that entire name ever again. I'm going to only say DMET. Otherwise, this talk will probably take like 30 minutes. So this work is driven by a need to expand on our current structure for clinical testing in the United States. The FDA does require preclinical testing in animal models for acute toxicity, but most of these models, as you can see in these graphs, do a pretty poor job at predicting toxicity in humans during their clinical trials. And what we believe is happening is that these preclinical trials are lacking an amount of diversity, either because they're being done on inbred animals or because the size of the study is extremely limited. So of course their diversity is going to be limited as well. So our collaborator, Mary Mosedale, has been really spearheading this campaign to use the collaborative cross in pharmacogenetics, both for being able to predict candidate risk factors and also testing in humans before they go into clinical trials. So in this talk, or what my lab primarily focuses on in this context, is using the collaborative cross for this purpose. The reason why we are choosing collaborative cross is because of its reproducibility, its rigorous documentation, the scalability, and most importantly, because it does allow for replicates, genetic replicates specifically, so we can have control and treatment groups. So our lab has been doing these types of studies for a while now. We've been able to identify several candidate risk factors for gene by treatment effects that are potentially causing toxicity in drugs that are failing out of clinical trials. And these studies have been extremely promising, and they've also opened up, demonstrated the need for further investigation and characterization of the collaborative cross at kind of a baseline level, which is what my work is on. So in this presentation, I'm talking to you guys about our characterization of genetic regulation of DMET pathways across the collaborative cross strains. I'm also hoping to kind of give a good example of a rigorous QTL pipeline that can maximize the amount of information that you guys can get from these types of data sets. Because these data sets can often take quite a long time to generate, they can be quite expensive, and you want to get the maximum amount of information possible. And this is just a quick overview of the data that I'm working with. We collected transcriptomics and proteomics from 4 male replicates from each of the 45 collaborative cross strains. Just kind of a caveat, male and female mice do vary pretty significantly in DMET pathway proteins. And so we're currently working on grants to expand these type of quantifications across both genders. Where we first start with this type of project is estimating the heritability, which is the role that each of these strains seems to play on the level of the protein expressed. And the DMET proteins exhibit a really high amount of heritability across all of the proteomics that we quantified. For the rest of this talk, I'm actually also going to narrow in a little bit more, and we're only going to be talking about CyP2C39. That is because this protein exhibits a really interesting distribution of data, where several of the strains have a near zero level or an unquantifiable level of the protein exhibited in the liver. And if we were to handle this data in a typical QTL mapping method, then the only significant loci that we would be able to determine would be the loci that's distinguishing the difference between strains that exhibit a dropout of protein and strains that have a continuous level of protein. So in order to handle that, we did a two-stage QTL model where we first ran a binary QTL. We assigned binary phenotypes to the strains, depending on whether they were part of this category that had a near zero level of phenotype or a continuous level of phenotype. After we did this, we just subsetted out the data so that we focused on the continuous level of phenotype for our second round of QTL. Our binary QTL was able to identify a significant peak on the chromosome 19 for these mice. And we can investigate this peak a little bit further by looking at the haplotype effects. In this specific circumstance, we have a really strong haplotype effect from B6. So most likely, nearly all of the mice that inherit B6 at this loci have that dropout of protein, which is something really interesting to consider for future studies because a lot of people use B6. Looking at our continuous phenotype, we are able to identify another significant QTL on chromosome 5. And we can, again, investigate this a little bit further, but the haplotype effects are a little bit more dynamic at this locus. And kind of the important takeaway to take at this point is if we had just moved forward with our standard QTL at the beginning, this chromosome 5 locus would not have reached a genome-wide significance level, and we likely would have just disregarded it. So at this stage, we're going to look a little bit more closely at that chromosome 19 peak. We're going to do that by doing a merge analysis, which is a model selection framework to pare down from our 8 haplotype normal linear model that we run to do a QTL. We do a model selection to choose which SNPs exhibited in the population best fit into the model. And then we move forward with those SNP distributions or the strain distribution pattern here. So this can be a little bit to take in. On the right side in the legend, all of these 0, 0, 1, 1, 0, if you guys haven't seen that before, the very first number, that first 0 or 1, is going to be AJ. The next number is going to be B6. If it's 0, all of the numbers that are 0 have the same allele as all of the other numbers that are 0. 1s are going to be a new SNP that was found at the locus, and then 2s are going to be another new SNP. So in this blue one, the only one that exhibits a different SNP is 129. And the primary takeaway here is that by doing merge analysis and running a SNP-based QTL instead of focusing on the 8 founder haplotypes, we can better power our QTL mapping. I also want to point out, so before we were looking at the haplotype effects where B6 had a really, really strong haplotype effect, across the majority of this region, B6 does not have an independent or a unique SNP that we've identified. And so I've gone ahead and emphasized the loci that have a unique SNP to the B6 in red that reached or improved upon our 8 founder haplotype. And so these SNPs are potentially ones that are more, warrant more investigation. So up until this point, I focused primarily on the proteins that we collected in pQTL analysis. This is just looking at the relationships between the genotypes and the phenotypes of that protein. We can expand on this information by incorporating some of our transcriptomics data, because we all know that genetics are not going to be the only thing that is causing variation in our protein or phenotype of interest. There might be some other things at play, like RNA, another loci that's interacting, other proteins, et cetera. So Greg and Wes developed a Bayesian model selection framework within our lab to run this mediation analysis. And you can find the link there. We're primarily interested in identifying any relationships between the genotype, phenotype, and mediators where the Y is non-reactive. So the phenotype cannot regulate the genotype or the mediator itself. And whenever we incorporate that assumption, we have 8 potential models that are 3 kind of markers can exist within. If we assume that all of these models have an equal probability of existing, then we're going to assign a prior probability to a uniform prior probability to this model selection framework. And we can use Bayes' theorem to update our assumptions on the model distribution or the likelihood of each of these models existing with the data that we have. When we do that, we generate a posterior distribution. And so in this case, the model selection framework would prefer the 2 models that have some degree of mediation based on the data that we have. So we ran this analysis on the CyP2C39 protein, the chromosome 19 peak, and all of the transcriptomes. There are about 20,000 across the entire genome. And we were able to identify 2 potential mediators that showed a strong indication or strong amount of evidence for mediation based on our model. These 2, sorry, these words are quite small. I should have made that larger. But these 2 are CyP2C38 and CyP2C39. Our darker green is going to be the model where a complete mediation is more likely. And the light green is the model where the partial mediator existed. So CyP2C38 is the transcript for CyP2C38 is likely to be a partial mediator of the protein for CyP2C39, while the transcript of CyP2C39 has equal probability of being a complete and partial mediator. And these results kind of make sense when we look back at the confidence interval of our peak. This region is heavily enriched in DMET pathway genes, including CyP2C38 and CyP2C39, which makes sense in a genetic kind of context because transcripts that are near each other are oftentimes competitive inhibitors. These transcripts also share similar functions and have similar codes. And so it's likely that they are regulating or sharing a common promoter, and it's worth further investigation. So in summary, we hopefully demonstrated a case for analysis of the collaborative cross in pharmacogenetics. I demonstrated one example of the QTLs that we were able to calculate. We've identified dozens of QTLs within this data set, but if I focused on all of those, it would be far too long of a talk. And hopefully we also demonstrated a rigorous QTL pathway to take your data through in any of your analysis to extrapolate the most amount of information that you can. And of course, kind of a center point of this entire conference has been the use of these broader resources and databases to share our data so that other people can access it. And since the main purpose of this project is establishing the collaborative cross as a model for pharmacogenetics, that is our next step. So I'd like to thank my lab. Ellen is here right now. She presented on Monday. And if you have any questions about any of the methods that were demonstrated in this talk, you're welcome to look them up here. These are all the GitHub links. You're also welcome to email me or, of course, Will himself. Thank you.